Alright, so let's go over the tutorial, what is biology? I'm going to highlight all the necessary pages and we're going to go through this fairly quickly. I'm going to leave the exercises up to you, okay? I might note one or two of them if something too long. So let's go ahead and move on. Page 2, you'll see your objective, and from here on out, I won't be going over objectives, nor will I be going over the intro that you see on page 3. Normally your tutorials will start out with an objective and then some kind of story that relates the biological idea to everything that you're learning. That's fine, you can read the stories, they're absolutely helpful, but I think you might be able to get it if I just go over the important parts. So I'm looking at page 4 now, they talk about how biology is existent in everything, and now they're going to get into the actual important parts. So the first page that you're going to find really useful is page 6. This is going to be on your mastery test, so we will go over this. The first thing I want to go over is I'd like to cover themes of biology. And there are going to be a couple of themes, you can see them all, but science as a process is your first one. Alright, so let's see if I can. There we go. On paper, here's science as a process. Alright, so science as a process really just means that everything in science is going to follow steps in order to get to where you're going. It's not just a poof, now we have an idea kind of thing. You have to follow things like the scientific method, you have to understand how research works, etc., etc. So you just have to understand that science is not just a sudden idea of anything, it's an ongoing process of learning. So we keep changing, That theme is the most general, and that's what we're going to start with. Now we're going to go over evolution. Evolution is something that you are all probably fairly familiar with. You've probably heard of Charles Darwin. You guys have probably seen the picture of where they have like a monkey that turns into a caveman, that turns into a human, all that kind of thing. So evolution is the idea that organisms change over time, which is true. You don't have to believe that we necessarily started from monkeys. That's completely up to you. But what is true is that all organisms change. So at some point or another, human beings have changed from the cavemen that we were to what we are now. Giraffes, there might have been one with a short neck way, way long ago, but that no longer exists. So evolution is really just the changing of organisms, the improvement, or maybe even the worsening. It all kind of depends. So evolution, again, is just that change over time. All right. Your third one is going to be energy transfer. This is really simple. We all have energy. That's how we move, that's how we exist. Without energy, you wouldn't be able to do anything. So what I need you to know from this is just that energy moves from one thing to another. Normally it moves from plants to animals, etc., but plants have to get their energy from somewhere too. They get theirs from the light, sunlight, that kind of thing. So energy transfer is really just the idea that energy moves from organism to organism, from thing to thing. Alright? So that's actually going to be a recurring theme later on. You'll see this more. Continuity and change is your fourth theme. Continuity and change. All this really means is that when we reproduce, so humans reproduce, and it takes two people and 50% of each person's DNA mixes together, you end up with a new organism. Generally, that new organism is different. Some people look a lot like their parents, and some people often do the same things as their parents. But when it comes down to it, there are some slight changes. The continuity part is that 50% of their DNA material is still the same as their parents. It's just a combination of a lot of different, or two different DNAs that causes this change. All right. So in science, things are continuous and they do evolve over time. You just have to understand that the fundamentals are still there. So it's really important to understand the basics of biology. If you understand the basics, you can apply it to things elsewhere. That way you can see those changes and whatnot. Okay? Next thing we're going to go over is structure and function. This is actually really simple. All structure and function means is that we don't necessarily worry at all about how to create things. Things are already created the way they are because they work. For example, my pen is shaped like this, it's round, it's kind of thin, etc., because it fits in my hand really, really well. If we had created a pencil that was, a pen or whatever, that was shaped kind of like my phone, this would be really inconvenient to write with, and it's not a natural way. It doesn't fit my hand like a pen does. So pens have this shape because they work better this way. Conversely, a cell phone is not shaped like a pen because if this were shaped like my pen, I would have a lot of trouble texting on this. Okay? So everything, and this includes not just our technology, but organisms have their body parts because they fit a certain function. For example, fish have paddles. Well, not paddles, sorry. They have fins that are shaped like paddles because it helps them cut through the water easier, whereas we have feet, which helps us stand our ground better. So structure absolutely relates to function. Things are generally shaped the way they are because they do something that they're supposed to do. All right? Regulation. This is called regulation, but there's another word that I want you guys to get used to. It's called homeostasis. And homeostasis really just says that our bodies have a balance in them. And that balance is useful and important and necessary for living. If your body is overheating, there is a mechanism that cools it down. If your body, that's called sweat, by the way, if your body has a fever, that is because there is an invader there. So you had some kind of bacteria, some kind of virus, and your body's trying to heat up to get rid of it. So that is a mechanism that leaves us in homeostasis. The body likes to be the way it is. Every time things change, it does a couple of steps in order to keep you the same. All right? This is going to be a general theme of biology, and it's actually going to be very, very important. So if you remember that everything has a balance, whenever I teach you one word, I'm probably going to teach you another word that happens to change up and balance each other out. Like they normally neutralize each other. Okay? So whenever you memorize one word, try and pair it up with another word because there's a likely chance that there is another one. In which case, by memorizing one thing, you'll have the other thing automatically. Okay? Another theme that we have is interdependence. This one's actually really easy to explain. All it is that people, organisms, animals, etc., all depend on each other. So as humans, we eat vegetables, and some of us eat animals and that kind of thing. So we depend on these other animals for life. That's what that giant food web is. Okay? Plants depend on the sun for light, that sort of thing. Everything depends on everything else. So that interdependence is how we go about life. If you were to eliminate all of the fish, cows, pigs, I don't know, whatever else people eat, gator tail, like I don't know, whatever. But if you were to eliminate all those things that people eat, we would be in much worse shape. Okay? So that is interdependence. And then the very last thing, and this one's going to come up in your mastery test if I'm not mistaken, technology. But not just technology, technology and society. So what I want you to focus on, push that up a little bit, there we go. Alright, what I want you guys to focus on is we develop all sorts of technologies. Like surely you can find it around. I'm going to show you my cell phone. If you reach in your pocket, you've probably got a phone in there too, or maybe it's in your hands and probably shouldn't be texting. You should probably be paying attention, but you know, whatever. You do your thing. But yeah, so we all have technology really, really close to us. The societal part is what we're concerned with, though. We can advance technology as much as we want, and that's really, really wonderful, and it makes our lives so much better. Like, don't get me wrong at all. I'm not against technology. But the one thing about it is there are always societal concerns or ethical concerns. And what this really means is, let's say I develop the ability to clone human beings. Society says that's great, that's fine, but what about those clones? Do we treat them as humans, etc., this and that? Society is the reason why we can't just do animal testing freely. Like you can't just pull a dog off the street and test your random drugs on it. You cannot do that. That is ethically not okay. So what you'll notice as we go through this bio class is that there are going to be a lot of discoveries that, you know, do this and do that, but there are societal issues as well, like animal testing, cloning, genetics, baby birthing, that kind of thing. Like there are a lot of controversial topics. So when you have to do your essays at the end of your class, make sure to keep these themes in mind because all of these themes make excellent essay topics. Okay? Alright, so we're going to go ahead and move on. Let's see. So you'll notice that on page 7 you've got a little exercise there. I will give you one thing just for the record. Actually, no, all those make sense, so we'll go ahead and move on to page 8. Alright, so page 8 goes over a bunch of different areas in biology. Biology is a very overarching topic. It covers a lot of different things. So what I'm going to do is I'm going to help you guys understand what these other items are. So if you think about it, biology is kind of this gigantic upper subject of importance, and it's going to break into a bunch of other things, and then those things are going to break into a bunch of their own things, etc., etc., etc. So it's going to keep branching and branching and branching. You don't necessarily need to know what all of them do. We're going to go over a couple just because I think a few of these show up in your mastery test. So biochem, let's see, bioinformatics, bioinfo, and we have things like biophysics, etc., etc. And there are actually a bunch more. I'm going to focus on probably just these three. Maybe I'll do sociobiology too. So let's talk about what these four are, just so that it's a little easier for you to handle the mastery test. Biochemistry is literally the chemistry of the body. So in your body, plenty of things happen, plenty of things change. Biochemistry is a study of why they change and how they change. Bioinformatics is just a study of biology from a information or statistics standpoint. So you're just establishing what numbers mean. So if I tell you that it's this percent likely that this happens and that happens, then gene A affects gene B with a 10%, whatever. So bioinformatics is really just using information and data in order to make biological conclusions. All right? Biophysics and sociobiology. I need to move this. Biophysics is actually not covering the whole page. Anyway, biophysics and sociobio are two more areas of our upper area of biology. Biophysics is really studying how things move and how things travel around in the body. So for example, the study of how our nerve impulses move around or how quickly our muscles move, these are all going to fall under the broader area of biophysics. And then sociobio this is actually going to fall under something called ecology, which you're going to learn about later. That's also a version of biology. So sociobiology is a smaller form of ecology where you're really just studying relationships. This is just how things relate to each other. So human beings eat fish. So part of our sociobiological study would be the relationship of human eating fish. That's an eating relationship. Okay. Cool. So those are just some of the areas. I'm not sure which one shows up in your mastery test. Let me check. No, we're good. All right. So now we're on to applied biology. There you go. That was the Plato tutorial. You guys probably hear it yourself. You're going to get a question on page 10. I'm actually going to. Oh no, you should know this one. So. Yeah, page 10 will be fine. Page 11 we're going to look at. Okay, page 11 is going to talk about the scientific method. Now that is important. We're definitely going to have to go over that. So we're going to start on a new page here. And get that lined up. Push it up a little tiny bit. Alright. So let's talk about the scientific method. There are a lot of different ways that you can learn the scientific method. They're going to go over them in their own way on Plato, but I think this will get you through. My scientific method might differ a little bit from Pearson's, but it'll be really close to like Campbell's biology scientific method. In any case, as long as you have the general idea, I don't think anybody will worry about how you know it. Alright, so let's go ahead and cover the scientific method just a bit. Step 1 of the scientific method is generally something that says something like observe. Because you've got to see something, you've got to learn something, you've got to hear about something in order for that question to show up. So you want to observe, and there's going to be a problem somewhere, okay? Whatever it might be. Your second step is going to be hypothesize. So after you've observed some kind of problem, you're going to want to hypothesize about how to fix it or what will happen. For example, I observed a bunch of monkeys at the zoo huddling up in a corner of the zoo cage room environment thing. Okay, so a bunch of monkeys all huddled up together. So I could hypothesize that they are huddled up together because it's cold. I could hypothesize that temperature has something to do with their huddling. All right. So your next step here is to experiment. Now, one thing to keep in mind is that your hypothesis has to be testable. Okay? If your hypothesis is not testable, so it has to be testable. Has to be testable. If it is not testable, you cannot move on to the experiment phase, in which case you are no longer doing science. To come up with a hypothesis that cannot be tested is just guesswork. You're really just saying things. It doesn't matter at all in science. So it has to be testable. To test our example, we have a bunch of monkeys in a corner. I think it has to do with temperature. I think they're cold. So what do? Turn the heater on. All right. So if I turn the heater on, the monkeys will do something, and I'm not sure what, but let's find out. So here's step 4. Step 4 is to analyze. After you've turned the heat on, you have to analyze your data. Did they spread out? Did they stay huddled together? Who knows? But if they spread out, that means that I was probably right. It doesn't mean I was necessarily right, but if I heat them up, I was probably right that they were probably just cold and they needed to huddle together for the heat. Okay? If, however, they do not move apart, that probably means that they're hiding something, they're protecting something, or maybe they're just like sitting together. Who knows? The point is, you have to analyze your results and then conclude. In science, we have to conclude and share our results. So we conclude that temperature does affect monkeys huddling together. So, if it's warmer, monkeys spread out and they spread apart and they went all around the zoo. That's a good thing. That's fine. That means that my hypothesis is probably right. My conclusion is great, and I share that. Even if I'm wrong, however, I can still say, all right, well, the monkeys did not move even though I turned the temperature up, so there must be another reason for their huddling up. In which case, I still share those results, and I go see if other people do the same thing. Just because I can do an experiment does not matter. Multiple people have to do this experiment for it to actually matter. Okay? One other thing in the scientific method is about step 4. So let's talk about step 4 a little bit more. Actually, here we go. What I'm going to do is I'm going to just pull this over this way. So our analyze, all right? When we analyze data, there are actually 2 kinds of data. The first type of data is called quantitative. Quantitative. All right? The second type of data is called qualitative. The difference between these two is that quantitative, I can count it. All right? I can count it. What happens is that anything I can count, let's say I wanted to know the temperature at which monkeys start to spread apart. That would be a temperature thing. I would just have to get a thermometer, and there you go. I can count, or I can apply numbers to my temperature. If I wanted to talk about how many monkeys move about, that is counting, or that is quantitative. All right. However, on the opposite side, qualitative would be things that I cannot put in numbers. These would be questions like the monkey's fur changed color. That would be qualitative because I'm describing color. Or the monkeys developed a smell. In which case, a smell would also be. These are often related to the 5 senses. Alright, so hearing, smell, all that jazz. Generally not taste. You generally don't want to taste things in the lab, but you get the idea. It has to do with the 5 senses. So if it's qualitative, it's probably these 5 senses, and you cannot count it. As soon as you can count it, it is definitely quantitative. Alright, so that is how scientific method and scientific research happens. So we're going to go ahead and move on. I'm going to have you guys move on to page 12 on your tutorial. That's fine. Page 12 and 11 are actually everything that we just talked about, just in more detail. So we're going to move on to, actually we're going to keep going 13 the same way. So 14, you'll notice that on 14 they're going to highlight all of these scientific discoveries, starting from Hippocrates, going to Leeuwenhoek and Gregor Mendel, these kinds of people. We're going to go into detail about some of them more in the future, so I don't need you to worry about it right now. However, one thing I will note is that I don't personally care if you know these people. I don't think they show up that often. I will highlight any people I think you definitely need to know for the EOC. Okay, so don't worry too much about page 14 for now. Page 15 is going to go over microscopes. Okay, this is going to be on your mastery test, so we will have to learn it. There we go. Okay, so let's talk about our 2 types of microscopes that they're going to go about in this tutorial. And these are going to be the light microscope. By the way, this light microscope, I know it's not talked about in your tutorial, but it has another name. It's going to be called the compound microscope. I don't really know which one they're going to use in your mastery test, but here's one of them. Alright, light microscope / compound microscope. And then there's the electron, scanning electron. I'm going to call it that. I think they call it just the electron microscope, but that's okay. Scanning electron microscope is going to be the other one. A couple things you're going to need to know is that with the light microscope, it is going to obviously use light. If that wasn't obvious enough. It's also great for seeing living things. Alright, and the reason it's so good for this is because it takes light and it just shines it through cells or through, living things and that sort of thing. And it magnifies a bunch of times. It magnifies quite a bit, but it has only moderate magnification. So I'm going to say it's pretty good. It does help us look at things. But again, its main strength is that it sees living things. Like that is a really, really nice factor because I can just watch things move around and watch cells, explode or be invaded by viruses. That's actually a little hard because viruses are small, but you get the idea. I can watch plant cells, lice, and that sort of thing. Alright, so scanning electron, the thing that they're different from here is obviously they use electrons, electron beams essentially. And they're going to give you can't use them to see living things because they can't catch them, essentially. It's too hard to follow them around this way. But what you can do is you can magnify a lot. So this has like huge magnification. Massive magnification. So you get really, really detailed images with your scanning electron microscope. Okay? For purposes of your test, I think this is all you'll need for what is biology. So we'll go over more in the next tutorials. So let's move on to 16. 16 talks about the same sort of thing. So on page 17, you're going to have a little exercise on your tutorial. It's actually really hard. I'm going to talk about why this one is real quickly, just so that you guys have it. A paleontologist is trying to study the DNA of a fossil from an extinct animal called X. He has formulated 3 hypotheses. Which hypothesis cannot be tested? Number 1 says X's DNA has a high proportion of protein. Which one cannot be tested? So how would we test whether or not it has a high proportion of protein? And you're probably thinking you could just count it or, weigh it or something like that. Yeah, you're absolutely right. We can count or weigh protein. So we're not worried about A. That one is testable. So we're going to move on to B. Protein content of X's DNA changes with its life cycle. Probably, we could just look, like as it gets older, we could just see whether or not the DNA changes, right? So you could just watch this one. This one doesn't even require counting, is watch. Third one, X's DNA consists of a single strand. Also easy to tell because we can just look at the DNA and see whether or not it has 1 or 2 strands. By the way, DNA is probably going to have a double strand because it's deoxyribonucleic acid. RNA has 1 strand. So it's likely true, but it doesn't change the fact that we can just count the strands, right? It seems like all 3 of these are right, but there's actually a right answer, and it's going to be the one in the middle. The protein content of X's DNA changes with its life cycle. Here's the reason why. Your animal is from a fossil, or your DNA is from a fossil, which is from an animal that is extinct. You cannot watch an extinct animal grow up because it doesn't exist. Alright? Don't worry about how tricky that one was. I just wanted to explain it in case it bothered any of you guys while you were getting it wrong. Alright, number 18. Okay. Last thing we're going to go over in this tutorial is how to read graphs and how to do metric system. This is going to be the longest part of this lesson, so I'm going to try and keep this video under 30 minutes, so I'm going to roll through it. If you have any questions, feel free to ask me. Okay? So let's go ahead and start with how to read a graph first. You'll notice that you have a table and a graph. Let's talk about a graph. Alright? They give you 4 steps to analyze data, which is actually pretty good. These are pretty decent 4 steps. First off is classify the data into appropriate categories. The main categories you need are these bottom ones. So you'll notice that this one is days since planting, whatever. And this thing on the left is the height. Alright, both really useful data. And days since planting increases that way, height increases upwards. Okay, so it always increases out and up. So your major thing is what by what? What are we talking about? So this is the height of a plant and the days that it's existed. If you think about it, you can probably already start to assume that the longer you let your plant grow, the taller it will be. It actually makes perfect sense. But the main problem that people have is they get really thrown off because they're not even sure where to start. Now you know where to start. It's this by this. It grows this high for this long. So you imagine that it would look something like that. You would think that the longer it exists, the higher it gets. So it moves upwards. So it's, I don't know, it existed 10 days, and here's 5 days. Here's 5 height, and here's 10 height. You would think that at 5 days it would be around 5 height, and at 10 days it would be even more. Okay? So you know your graph is going to go upward like this. Looking at your example on page 18 of the tutorial, you'll notice that they have plant 1 and 2, and that they're both going upwards. That's exactly what we'd expect. We'd expect a plant to grow, right? So now let's draw an inference from the data. Looking at our two plants, you'll notice that you have like plant 2, which looks, whatever. I'm going to make this one plant 1 because it's kind of close. I'm going to make this one plant 2. Actually, it should still be going up a little bit. So here we go. So here's plant 2. So this one looks kind of like the one in your tutorial in case you're not looking at it. But the idea is this. Plant 1 is increasing like this. Plant 2 is increasing like this. So you'll notice they're both getting taller. There's no doubt at all. But if you look at these two, this line's higher than this line at every point. That must mean that plant 1 is growing faster. So our inference based on this data is that plant 1 grows faster. Okay? If you look at your table on that same tutorial page, they actually have this entire graph in numbers. Anything that I draw on a graph, I can absolutely put on a table. Alright? So if you look at that table, you'll notice that plant 1 is compared to plant 2, and they both start at 0 for the first 3 days. But then you'll notice that on day 5, plant 1 is 8 inches, plant 2 is 7 inches. It's 1 inch bigger on day 5. On day 7, plant 1 is 15, plant 2 is only 8 inches. So plant 1 grew like 7 inches, plant 2 only grew 1. So plant 1 must be growing at a higher rate. Okay, growing faster is all that really is. So this is how you analyze data by drawing an inference, and then you looked at the appropriate values, and then you drew a conclusion. Plant 1 grows faster. Okay? Now, we wrote it out step by step, but honestly, you could probably get through this all in one kind of fell swoop. You just have to get used to using that process. Alright? Next thing we're going to go over is metric system. Metric system is actually really, really important. I prefer it to the English system. I have a lot of trouble with the English system. Metric system is all based on 10. So every time I do anything in the metric system, it's all going to be based on the idea that I'm moving the decimal left or right some number of places. I just have to figure out how many places. So one of the main issues is that people have trouble knowing how far to go. So in the metric system, you have to learn your prefixes for all of your words. Prefixes are those little like part of words attached to other words. So for example, you guys know what a meter is. Alright? So a meter is going to be our basic unit. This is just 1 meter, whatever. Meter is going to be our root or our base. Okay? So this is what we're going to work on to learn the metric system. It's just all based on 1 meter. Alright? 1 meter is equal to a bunch of centimeters. Alright, and you probably have an idea of this. Centimeters are smaller than meters. It's kind of like I have a bunch of M&Ms in a bag, and the meter is my bag. So the meter is 1 meter, 1 bag. But if I change that into individual M&Ms, I have to open up my bag. So my 1 bag is equal to a bunch of M&Ms, right? So I know that this 1 meter is going to be equal to a bunch of centimeters. Centi comes from the word century, or rather it has the same root as the word century, and century is 100 years. So the centimeter is just 1/100th of a meter. That must mean that this 1 has an extra 100 centimeters. So I'm just going to take my decimal and I'm going to move it over twice, because in 100 there are 2 zeros. Alright, so I'm going to move over 1, 2. So a centimeter, I'm sorry, 100 centimeters is equal to 1 meter. Okay? On that same token, let's say we want to go bigger. So 1 meter is equal to how many kilometers? Now, you guys have probably heard of kilometers. Kilometers are kind of like the European / every other nation in the world other than America's measurement of distance that's like a mile. Okay? So kilometers. How many kilometers are in a meter? Now, this is a funny question because here's the thing. I had a bag of M&Ms, right? So this is my bag of M&Ms. But this kilometers thing is like my shipping package from a company like UPS or whatever. And they're shipping like a whole case of M&Ms that has a bunch of bags in it. So one bag is not equal to one case of M&Ms, or just like 1 foot is not equal to a mile, or that kind of thing. Okay? So to change 1 meter into kilometers, we had to figure out the conversion again. Here's kilo. Kilo means 1,000. And if you look, there are 3 zeros instead of the 2 zeros. All right? I know kilometers are bigger than meters. That must mean that I need to move my decimal a different way. So if I go from larger to smaller, I'm going to increase my number of zeros. I'm going to move my decimal to the right. Okay? So to the right is what that was. Now this one here, though, I'm going to move my decimal to the left because I'm getting a bigger unit here. I'm going from 1 bag of M&Ms to like a portion of a case. Like my 1 bag is 1 something of a case. All right, it's a tiny, tiny part of a case. It's kind of like I have this cup, and this cup can fit 4 pens. Just pretend like 4 pens is the maximum. But I'm asking to change 1 pen into how many of a cup? This 1 pen is 1/4 of that cup, if I can fit 4 total. Alright, that was my drinking cup, so that was kind of a waste, but hopefully that'll make sense. So, we're going to move this decimal to the left. And we're going to move it to the left how many spots? The 3 zeros. We're going to move it 3 spots. I'm going to go 1, 2, 3. It's kind of sloppy, but that's okay. 1, and so there's our new decimal. Our 1 meter is equal to 0.001 kilometers. Okay? So no matter how this works out, I'm just moving my decimal left or right in some way. I'm either moving left or I'm moving right, and I'm moving however many spots I need to. Centimeters was 2, kilometers was 3. I'm going to show you guys another one in just a sec. But this doesn't matter if I call this a different thing. It doesn't have to be a meter. I can call this a gram. I can call this a liter. I can call this whatever, as long as it's metric system. The metric system is all about moving your zeros. All right? Unlike, of course, the English system, because like English system is 1 mile is equal to, so a mile, let's compare it to feet, right? So it's 5,280 feet or something like that. That's ridiculous. Like why? Why would you want that? That's terrible. It's so much easier to move my decimal around. Like if I wanted to change one, here, let's do an example. Let's change 3,241 meters into kilometers. All right, equals how many kilometers? We don't know. All right, it's pretty simple. To be able to change this over, all you're going to have to do is you have to move your decimal. So let's take a look at last time. When we went from meters to kilometers last time, we moved our decimal over from over here, 1, 2, 3 spots to the left. So I have meters now. Let's move it over. 1, 2, 3 spots to the left. So my answer is 3.241 kilometers. Okay? So it's 3 point, let me make that decimal a little bigger. There you go, you guys can see that. So it is a decimal over here. So you ended up with a smaller number, and that makes sense because it takes a bunch of these to make up 1 kilometer. Okay? So that is how metric system works. So I know I went over a little bit. We hit 33 minutes for this video, and I apologize for that. But hopefully this all helped you out. This is going to be a foundation. I'm going to work on getting these videos a little shorter. And I'm going to hope to narrow it down a little bit better. But if you guys have any questions, feel free to message me. You can email me, it's at the top of the syllabus, or you can always message me on Play-Doh. Check out my website. Check out this video on YouTube. Wherever you need to, just let me know what your questions are, and I will get back to you as soon as I can. Alright guys, good luck, and good work. Let me know how you did on the mastery test. Don't stress yourself. If you have to go back to the tutorial, go ahead. Feel free to watch this video, pause, do whatever you need. Alright guys, let me know if you need anything.